My name is Ralf Paus. I'm a research professor of dermatology at the University of Miami Miller School of Medicine, and I've worked for a long time on hair research. In addition, we have investigated neurohormones of human skin, and in both areas we have done some pioneering studies. And this is where the hair follicle enters the picture. This tiny but miraculous mini organ that makes hair shafts, for example, on your scalp. This organ is so special because it not only is highly responsive to numerous hormones which control its growth and hair shaft production and hair pigmentation, but it even makes these hormones themselves. And the discovery that we made about 20 years ago is that one of these hormones that is hardly ever talked about in hair research is prolactin. Prolactin is most famous for being the key hormone that controls milk production. That's where the name comes from, prolactin. But we now know that this hormone does multiple other things. It is controlling growth, it has immunoregulatory functions. And about 20 years ago, we found that not only do human scalp hair follicles respond to stimulation with this hormone, in fact, they were inhibited when we microdissected and organ cultured them by prolactin, but they even made prolactin themselves. And this is where ABS-201 comes in, the prolactin receptor blocking antibody developed by Absci. The company had read this old study and thought, well, could we perhaps promote human hair growth by blocking the receptor for this neurohormone in order to keep the hair follicle longer in its growth phase in Anagen, and thereby reduce the daily hair shaft shedding and get longer lasting effects than you would get with normal drugs with a short half-life. And we used a system that we have been using for many years to study the effects of all sorts of hormones and neurohormones on human scalp hair follicles. What you can do is you can microdissect these little organs and then you throw them into a culture medium, and crazily enough, they still grow as if they were on the living human scalp. And then in this assay, as shown here on this slide, you ask the question, is my test substance, in this case, for example, prolactin or the prolactin receptor antibody, is that pushing the growing follicle, the Anagen follicle, and you start with 100% Anagen follicles in your culture, is that pushing the Anagen follicle faster into the regression phase, catagen, or is it retaining it longer in Anagen? And if it retains it longer in Anagen, that means you will have less hair loss. So what we are really looking for in this assay is, on the right-hand part of the slide, the transition of Anagen follicles into catagen in this hair cycle. But in this case, we studied it not in isolated, microdissected hair follicles, but we took the entire human scalp skin. And specifically, we used so-called temporofrontal scalp skin. That is the scalp skin where androgenetic alopecia first becomes manifest in males. And why did we take the entire scalp skin and not microdissected hair follicles as we had done 20 years before? We did that because we know that many cells in the skin have receptors for prolactin and can respond to it. And we wanted to see how this antibody is impacting on hair follicles that are living in the real tissue environment that they see in the living human body. And what you do then is you take photos, you see this on the right-hand side, of scalp skin biopsies that you get from volunteers, and you see what is changing in the hair shaft production over 6 days. And then you take sections of these tissue fragments and you look at them under the microscope and you assess most and foremost what is happening with the hair cycle. Are these hair follicles staying longer in anagen, or do they go faster into catagen? That's the key question you have. And then you can look at all sorts of additional markers that get you a better idea of what exactly is happening. The first thing you need, of course, to check in this system is, does this antibody even do what is claimed that it does? And if you want to show that your antibody, in this case, is blocking signaling through the prolactin receptor, you have to show that phosphorylated STAT5, one of the signals that transmit a signal after stimulation of the receptor by prolactin, is upregulated. And you see this in the green bar on the right-hand side, that's exactly what happens. If you stimulate scalp skin with prolactin, then you get an upregulation of this signal. And if you do this in the presence of the blocking antibody, this is abrogated. That was good news, so we know this antibody is really active. It does what it's supposed to do at the level of the prolactin receptor. And this perhaps is the most important slide that we initially, the most important result that we initially got. So we looked, was there any effect on hair shaft production? And as expected from our older studies in microdissected hair follicles, if we stimulated entire scalp skin, male scalp skin, with prolactin, hair shaft production over 6 days was reduced. And the follicles, the number of follicles that stayed longer in anagen in the growth phase, you see this by photo documenting that the hair shaft gets longer, was actually increased not only when you co-cultivated prolactin with the prolactin receptor antibody, but even by the prolactin receptor antibody itself. That's the bar on the second bar from the left in the center. So that this antibody alone has already hair growth promoting activities suggests that it is actually inhibiting prolactin that is locally made in the hair follicle, just as we had published many years ago. And I was particularly interested to see this result and quite gratified, because when we had first published this, there was lots of disbelief in the field that this actually is real. But this is all macroscopic data that we see, so what you can almost see with the naked eye. But the real litmus test is to use histological, microscopic markers to prove that these follicles are really longer in anagen, and that the antibody against the prolactin receptor can antagonize the push towards catagen that prolactin gives the follicle. That's exactly what you see on this slide. So again, the antibody alone, the second bar from the left, keeps hair follicles longer in its growth stage. If you only give prolactin, they move into catagen, and if you give prolactin and the antibody, you again have an anagen prolonging effect. And then you can look at this more fine-tuned, and you can see what is the effect on cell proliferation and cell death, apoptosis, in the actual hair shaft factory, which is the so-called Anagen hair bulb, the Anagen hair matrix. And no surprise, what you see here is a phenomenon that perfectly supports what we had seen in terms of the hair cycle changes. Proliferation in the hair matrix is stimulated by this antibody against the prolactin receptor, and cell death, apoptosis, is reduced. And note on the graph on the right-hand side, the blue bar, the impressive cell death protection effect of just the prolactin antibody, prolactin receptor antibody itself. When we initially started with this work and we discussed how to set up these experiments, I reminded our colleagues that 5 years after our initial paper, we had found in female scalp hair follicles, and if you blocked the prolactin receptor with a drug that may not have been as selective as this new prolactin receptor antibody, the stem cells did not like it and went into apoptosis. Therefore, we were nervous to see what would happen here in male scalp hair follicles in the stem cell compartment, the so-called bulge. And lo and behold, the stem cells loved it. Their apoptosis actually went down when they were stimulated with this prolactin receptor blocking antibody. In the presence of prolactin, the stem cells even proliferated, divided more than in the vehicle control group. That was excellent news and is particularly interesting in a context of androgenetic alopecia, because androgenetic alopecia has two driving factors. One probably critical driving factor is that the follicle gets smaller and smaller due to missing inductive signals from the so-called dermal papilla at the base of the hair follicle. But the other contributing factor is that the capacity of hair follicles to generate daughter cells from these stem cells is diminished. So we looked at that too. And interestingly, if you just give prolactin, that's the green bar on the left-hand side of the slide, the number and the expression of these cells that are daughter cells that are directly produced from these stem cells gets reduced. And if you give the prolactin receptor blocking antibody, this can be completely reversed. That is fascinating from an androgenetic alopecia perspective, because so far we have no drugs that can really do that. So it appears that just blocking signaling through this one neurohormone receptor can restore the capacity of hair follicle stem cells to make daughter cells, and thus keep the organ in its desired large size. But that's not all. When I first saw these slides, I said, well, God, what is this antibody not doing? So it's actually improving the production of the key growth stimulating growth factors that the hair follicle makes itself. If you dump prolactin on male scalp skin, the green bars, then the production of key growth factor called insulin-like growth factor 1, IGF-1, goes down, and even that of another important growth factor, FGF7, fibroblast growth factor 7, goes down, and that can be completely counteracted by adding the prolactin receptor antibody. But even better, insulin-like growth factor 1, the one growth factor that in human hair follicles keeps the follicle longest in its growth phase, is stimulated just by the antibody alone. That means that just blocking the prolactin receptor with this antibody increases the capacity of the hair follicle to produce growth factors itself, with which it maintains and promotes its own growth. Yeah, and finally, we had shown before that, you see this here again on the left-hand side of the slide, that hair shaft production is stimulated by adding this antibody. And if you see this macroscopically, you would expect that also the production of hair shaft keratins, proteins that make the hair shaft, is upregulated, and that's what you see in the graph on the center, and in the immunofluorescence microscopy images on the right, that's exactly what happens. So the antibody has the capacity to prolong the growth phase of the hair cycle and thereby reduce the daily hair shaft shedding that dermatologists call telogen effluvium. But it moreover also promotes hair follicle stem cells themselves, and specifically their capacity to produce new daughter cells and to maintain this. This would antagonize the androgenetic alopecia associated hair follicle miniaturization process. And as if this were not enough, the antibody also stimulates the follicle to make more potent growth factors itself and to increase hair shaft production. Very impressive. I did not expect that when we started this. In reality, I was fairly skeptical that any of this would happen, but it did. And we repeated this in 3 donors, and that's about the time point when you feel confident about your own data. So in summary, we're looking here at an antibody that really blocks signaling through the prolactin receptor directly in human scalp hair follicles, in intact human scalp, so in the target organ that we want to treat clinically. And it does this on multiple levels. It, on the one hand, keeps the follicle longer in anagen. On the other hand, it stimulates the production of growth factors that the follicle needs to remain in anagen. And finally, it even stimulates stem cells and their capacity to generate new daughter cells, and also promotes hair shaft production. That a single neurohormone receptor antibody could do all of this is highly unusual. I did not expect it, and I'm quite impressed by this. Until now, we discussed our data from organ-cultured male scalp skin. But now it's time to talk about clinical trial data, where we go from ex vivo to in vivo. And this will be introduced by Dennis, followed by Professor Rod Sinclair from Melbourne.